what is referred to as TMDL effective monitoring plans. So TMDL stands for total maximum daily load. It is used extensively, really for about the last 20-25 years, to determine the maximum pollutant amount that a body can receive without exceeding water quality standards. So prior to this, monitoring was done much more on a concentration basis. The change was to regulate discharge on a mass per day and to take into account both point and non-point sources. So by point sources, we're talking about the wastewater treatment plant effluent. We're talking about a discharge from an industrial facility. It could be cooling water from a power plant. Non-point sources include everything from stormwater runoff, agricultural runoff, can even include the releases from contaminated sediments. And we're looking at how that impacts water quality. And using that, then we can develop management strategies that are really focused on the problems related to that body of water. And we'll look at a few examples that will hopefully help understand what this whole process really looks like. So, as I mentioned, what the goal of this is to identify water quality improvements, or the lack thereof, that result from some sort of implementation. So, for instance, the East Lansing wastewater treatment plant facility just switched from chlorine to UV disinfection. And as we'll talk about, the red cedar water is impaired with regard to E. coli bacteria. So there's extensive monitoring that has to be done on the Red Cedar River. So question that appropriately is asked now is, how will that water quality be affected by this change in treatment? Williamston is looking at increasing the capacity of their treatment plant. Again, change is being proposed that will ultimately be implemented, and then we want to be able to use a strategy that is appropriately designed in order to determine whether or not we're seeing any changes, hopefully improvements in water quality. And then this provides a feedback loop. So the state is the one agency, so in our case it's EGLE, has primacy, and they then determine what these TMDLs are for a treatment facility. So then the question is, if there aren't improvements, what needs to change? Do they need to reduce the limits? Basically, what's typically done then is one would review the existing data and information. We would select sites and what parameters are to be measured, design a study, estimate the sample size, and then develop the effective monitoring plan. And all of this is done to give this quantitative measure of the attainment of water quality standards. It just says the same thing, just in a another set. So the goal, like I said, is looking at initially all the data that exists. We didn't do that for the lab. We didn't have much data that I had available to look at phosphorus levels, but if we had, and we were using the information and basically doing a TMDL monitoring plan, we would have looked at that data before we determined any sites that some of you sampled on the Red Cedar River. And then I selected sites purely based on what I thought was accessible. And that is part of what is done. But also, I also chose the parameters based on what kits you had available. In reality, you would be looking at what parameters are relevant to that question that you're asking in terms of assessment. So we'll look at each of these steps in more detail. Typically, what is done is what is referred to as a watershed approach. These are the approved watersheds for the state of Michigan. So they're approved by DEQ. And you can see that they vary in terms of the issue that's being addressed. So if we look here and we're looking at the Bell River, that is addressed for dissolved oxygen. If we're looking here at Pine Creek, Upper Maple River, that is addressed by, what's addressed is the nutrients. So there, the concern is the level of nutrients in these water bodies. So they're impaired in terms of nutrients. So you'd be sampling for nitrogen and phosphorus. In the case of the River Raisin, nitrate is of concern. So their nitrate is your monitoring plan centers around nitrogen or nitrate. For the Grand River, it's a combination of dissolved oxygen and the other is E. coli. And we'll look at the monitoring plan for the Red Cedar River. So the goal here is not to look at the river in isolation, but to look at the river in the context of that entire watershed. So you're looking at the landscape. What are the land uses? What are the ecological resources? What river or water bodies are connected? And how do those connections affect water quality? The benefits of doing this is that you can do a much broader assessment of the pollutant sources. You can capture interactions between upstream and downstream sources and their impact on water quality. Because you're looking at the system comprehensively, that actually reduces the cost. You're also involving multiple water stakeholders. So you're looking at involving the stakeholders in that watershed. So these may be farmers, these can be urban landowners, industries, wastewater utilities, could be water utilities if they're using surface water. And then you can integrate these into other watershed programs. So you're integrating the whole TMDL monitoring plan into other watershed programs. So when you're looking at determining your sites, there's a number of approaches that you can use. This is referred to as the pour point method. So this is your watershed. This is the boundary of your watershed or the divide. These are the main water bodies and the tributaries. and you're monitoring at the outfall. So for instance, if we were looking at the Grand River and we were using this approach, where would we monitor? Anybody? Anybody know where the Grand River empties into? It's Lake Michigan. So we would be monitoring at Lake Michigan. So that's basically we'd be one set of samples, and our monitoring site would be at Lake Michigan. The distributed sampling method, you would be sampling both the tributaries for that main water body. So for instance, let's say this is the Grand River. This is the Red Cedar. Red Cedar flows into the Grand River, so we would be sampling in the Red Cedar. We'd sample in Grand River. We would probably sample on Sycamore Creek. So there are a number of water bodies that we would sample in that watershed. So our sampling is distributed over that water sample, over the watershed. The sites that we pick are based on where we would expect to see discernible water quality effects. So for example, if we were to look at the TMDL monitoring plan for E. coli in the Red Cedar, you'll see that they sample up in Williamston. They sample upstream of the Williamston wastewater treatment plant. They sample downstream. So you can look at the impact of that facility on water quality. Same thing with the East Lansing plant. You can also look at the potential for illegal discharges. So where are potential sources? Where are industries that could be potentially discharging? Those could be closed facilities that perhaps have a groundwater that's contaminated, and that contaminated groundwater is seeping into the river body. So this is why we need to know about the land uses so that we can determine where to sample. It can also be non-point sources. So what, for instance, are the agricultural operations in this area, and where are the runoff likely to impact the river body? Are there many stream channel restoration projects? There's been recently a few near campus or on campus where there's been some channel restoration. And how does that impact river quality? And then the now improved on-site wastewater management. So here you're typically talking about a more rural area that was, for instance, on septic systems, but because of the contamination or the increased density of those homes, potential contamination of groundwater, perhaps they've put in some sort of small on-site wastewater treatment for that maybe 20, 30 houses. So you want to look at what impact does that have now on water quality. So you would choose sites accordingly based on that. So in terms of parameters, it really depends on what you're attempting to measure. So for instance, common pollutants are total phosphorus, total nitrogen. That nitrate impacted, then maybe we want to break that down to total nitrogen. We'll also look at ammonia, nitrate, and nitrite. Look at total suspended solids, E. coli, commonly dissolved oxygen, conductivity, pH, temperature, turbidity. Notice these were a lot of the same parameters that you've been measuring this semester. They're similar parameters to what you measured when you did Lab 3 and you were sampling in a body of water. You also want to look at stressor and response variables. So you may also look, for instance, at issues like invertebrates. What types of invertebrates are present? And that gives you an indication of river quality or water body quality. And then covariates, this is often things like precipitation and stream flow. Because if you have increased precipitation, you'd expect to have increased runoff. And that may result in, for instance, increased E. coli levels. It may result in increased suspended solids or increased nitrogen phosphorus levels if you have nitrogen and phosphorus applied to a land. So this figure here is from the TMDL for E. coli in portions of the Red Cedar River and the Grand River. And this looks at river quality from around Weberville to just west of Lansing. So it incorporates about 40 miles of river. And it looks at both tributaries and the main river, which is the Grand River. And here's a plot from there looking at E. coli levels. So it's measured as colony forming units per 100 milliliters. And it is a function of time. And these are various different locations along the river. And the G stands for the Grand River. So there are locations along the Grand River. And what you see, actually, let me ask you, what do you see when you look at this graph? Any observations you can make? Ready? So the levels tend to be fairly low, but they increase significantly during rainfall events. So that tells us that there's a significant source of E. coli in runoff. Now, it doesn't tell us whether it's urban or rural, but it tells us that there's a relationship between these E. coli levels and runoff. We can also look at some additional data here, and this looks at the data under different flow conditions. So what it numbers of E. coli per day. So it's based on, it's a daily value or value integrated over a day is what it is. Okay. And it's under high flow, moist conditions, mid-range conditions, and dry conditions, and low flow. In an attempt to model this data. And then they compare the data to the levels for total body contact, that's TBC, and partial body contact. So for instance, total body contact is swimming in the river, partial body contact is kayaking in the river. And So Grand River is designated impaired with regard to E. coli. And the water quality standards were promulgated under Part 31 of the Water Resources Protection of the Natural Resources and Environmental Protection Act and REPA. The total body contact is monitored from May 1st to October 31st, and partial body contact is year-round. Total body contact is a 30-day geometric mean of 130 E. coli per 100 ml. So if we go back and look here, okay, so it's a 30-day average at 130. Okay. This is the partial body contact right here. With a maximum of 300 E. coli CFUs per 100. The partial body contact is a maximum of 1,000 E. coli. The wastewater discharge is not more than 200 fecal coliform bacteria per 100 mL over a 30-day period, or not more than 400 over a 7-day period. So not more than 100 fecal coliforms per 100 mL over 7 days. So one of the things you will do is, I've got real data for E. coli in the Red Cedar River. We've got data from about 2005 to 2020. And I've picked certain years for you to take a look at. And then I have data from the East Lansing wastewater treatment plant from 2007 to 2008 that you will also look at. And what you'll be doing is comparing this data to these TBCs and PBCs levels. Now, given what you see here, next time we have a high flow condition and you see a kayaker at the rapids, what recommendation would you make to that kayaker? Anybody want to comment? Let's put it this way. I wouldn't want to be in that body in the Red Cedar, because that's when we see the highest levels of E. coli. When we're looking at designing our study, we can look at a number of different ways to evaluate the data. So, for instance, we can look at a before and after. So the first one was that poor model, where now you're just looking at the outfall. So for instance, the outfall of the Red Cedar in Lake Michigan. And we're looking at before and after some significant change, some significant improvement, for instance, in a wastewater treatment plant, or significant changes in preventing or treating stormwater runoff, agricultural runoff, etc. The second is to look at an upstream downstream. So for instance, looking at the new or the changes in the East Lansing wastewater treatment plant, and what impact does that have on E. coli levels and changes. And then that feeds back to what our TMDL should be for the wastewater treatment plant. You can also look at a paired watershed. So you've got two very similar watersheds, and you're looking at, it's really not a before, it's, you've got a control and you've got a treatment is what you're really looking at. And then you can look at trends over time. So how is that water quality changing over a period of time? Sampling that we did was one point in time. If we repeated that later in the semester, we could look at changes. If we kept that data and looked at it again next year, and the following year, we could do some trend monitoring and determine how is that water quality changing over time. Now, to estimate sample size, typically what one wants to do is to determine the number of samples needed to demonstrate a statistically significant change. So if we're looking at a 95% confidence interval or an alpha of 0.05, then we're going to look at the number of samples needed to demonstrate that change. When I had you look at samples, I just determined number of samples, what I thought was reasonable for you to go out and sample. I made no attempt to do any sort of statistical analysis to determine that. And what is often used is what's referred to as a power analysis, and we'll look at that. But that takes into account the significance and then the standard deviation of the method you're using. So you need some sort of pilot data, and then you can use that then to determine the number of samples. So this is the equation that's used. I'm not going to go into any detail. You're not going to need to use this, but just to give you a sense of kind of how it's used is, here's a graph, and you're looking at, for in this case, the minimum detectable change in E. coli levels per year. So if you're looking, for instance, at 30, that tells you that you need somewhere around 60 samples. And there's graphs like this, and typically one would work with a statistician in order to determine this. Let's just look at an example here. And we have a situation where Fox Run watershed is impaired with regard to total phosphorus. So perhaps we've got this agricultural operation applying manure to land that we have significant runoff from the agricultural land. And so our TMDL is for total phosphorus. So what we're going to do is we're going to monitor for total phosphorus. Since often nitrogen levels are often elevated for the same sort of runoff, in the same sort of runoff, we would do nitrogen. It's land runoff, so suspended solids would make sense. It is an agricultural operation, so monitoring for E. coli. And then you have a potential for organic matter being discharged, so we want to monitor for dissolved oxygen because we want to determine whether it's being depleted. Conductivity gives us a sense of the changes in water quality, and then pretty much always measure for pH and temperature. If you're going to do pH and temperature, conductivity is easy to do, as you know. And then often turbidity, that gives us a sense of how clear the water is. Once we start doing this, then getting this data, then we can start looking, for instance, at correlations. We can start analyzing the data. So for instance, we can determine whether or not there's a correlation between total phosphorus and stream flow. Based on this data, it looks like there's a strong correlation. Notice here these are both in a log-log plot, as shown here. You can also do a correlation matrix. Back to assignment 1, looking at the correlation between chloride conductivity, total nitrogen, total phosphorus, total suspended solids, turbidity, and the other parameters. So you can see there's a positive correlation between conductivity and chloride, total nitrogen and chloride, strong correlation between conductivity and chloride, not surprisingly, fairly strong correlation between conductivity and total nitrogen. Let's look at total nitrogen and total phosphorus. I would have expected a stronger correlation. It's fairly weak correlation there. But it gives you a sense of these correlations. Strong correlation between total suspended solids and turbidity, I'd expect that. You can look at these correlations. You can also look at the data and develop a histogram. So you can see on the left is plotted total, it's the log of, actually let's look at the one on the right first. The total phosphorus versus frequency. Okay. One of the things I want you to note here, so this is the number of samples at 0.01, number of samples looks like at 0.025, but it's number of samples at various different concentrations. I want you to note here that what you have is a skewed distribution. So it isn't normally distributed. All of the statistical tests that we looked at earlier in the semester, that we've used throughout the semester, are based on a normal distribution. Because these are not normally distributed, you would have to use what we refer to as non-parametric tests in order to evaluate the data with any of the statistical tools. However, you can do a log transformation. And when you do a log transformation, notice that what you see is the data looks much more normally distributed. So in this case, you could use the statistical tests to evaluate the log of total phosphorus, even though you could not use those same statistical tests to evaluate total phosphorus. So in many cases, we do data transformations in order to use the statistical tests, what we refer to as the parametric statistical tests, for data that is normally distributed. They're much more powerful. We can also do time series plots. So you can see here the data, this is for fecal coliforms, and the data is plotted as a function of time. We can also do box plots. And in this case, we actually are providing the reader with some additional information. And these are just done, for instance, comparing spring, summer versus fall, winter. But notice what we can observe from this is the median. We see the 75th percentile, the 25th percentile, minimum and a maximum. So it gives you a more comprehensive view of the data. So when you're looking at selecting the test, the first thing you need to do is determine whether or not your data is normally distributed or not. If it's not normally distributed, can you do a transformation that would allow it, in which case it would be normally distributed? Typically, that's an inverse or a log. Are there outliers? The other thing is, are there data that are below the detection limits or above the quantification limits? That becomes difficult because then if you just simply put, for instance, above, let's say the limit of measurement is 1,000, and you analyze the data with 1,000, that's fine if the data is 1,050. If the data is 10,000, that will significantly skew your results. If it is below detection limits, often what we do is use either 1/2 or 1/3 of the detection limit in the data analysis. It's actually a little bit easier with below detection limits than it is above detection limits. We do not report data as 0. Something I should have mentioned earlier, but I'll mention it now. Okay, why don't we report data as 0? We simply don't know if it is 0. If the detection limit is 1, we don't know whether it's 0.9 or 0.009 or 0.000 the gazillion zeros 9. All we know is it is less than 1. So as you proceed, think about that in reporting your data. What is that detection limit? And how should you really be reporting the data? We also often want to look at seasonality. So do we see changes with season? And then we often have to deal with missing data. How do we deal with missing data? In Excel, if you use a hatch mark N/A, then Excel knows that there's missing data and it won't evaluate that cell as 0. So that's a useful tool in evaluating data. The next thing you want to think about is what is the appropriate test to use. So for example, if you're comparing 2 independent sets of data, so this would be before or after some remediation or mitigation strategy, an upstream or downstream. So those data sets are independent. If your data is normally distributed, you can use a 2-sample t-test. If your data is not normally distributed, then you have to use a non-parametric test, and then you'd use a rank sum test. If you're comparing 2 sets of data with matched pairs, so you're looking at pairing watersheds, upstream, downstream, then you would use a paired t-test. If the data is normally distributed, a non-parametric test. If it's not normally distributed, then you'd use a non-parametric test or assigned rank test. If you're looking at the relationship between one data group and time. So we're not looking at seasonality, we're just looking at time. Then you're doing some sort of trend monitoring. So how does the levels of E. coli, for example, change with time? If your data is normally distributed, you use a linear regression. If it's not, then you use a Kendall test. The only, as I mentioned, the only thing we'll talk about here are the parametric tests. If you're looking at seasonality, then you're looking at trend monitoring. Again, you use linear regression, but you'll use a seasonal term rather than just time. If the data is not normally distributed, then you use a seasonal Kendall test. There is a TMDL effectiveness monitoring tool. It's provided by EPA. It's Excel-based. It actually has some enhanced features with VBA code. And the purpose of this is to enhance planning. So it actually allows you to create some exploratory plots. It allows you to estimate sample size. It even allows you to estimate monitoring costs. So for example, I've provided the tool on LonCappa. You could download this. You could input the data from Lab 3, define your study, and actually determine the number of samples. It would give you a sample cost. It would give, so you could actually take that data as the pilot data and then use that data to design a more comprehensive study. So any questions? You're all very quiet.